Among all the virus infections, we put them into two general classes. There are acute infections and there are persistent infections. And I think from the names you can tell what they're like. Acute infections we'll talk about today. Those are the ones that come and go. You resolve them and hopefully you're alive and go on with some immunity. Persistent infections last for the life of the host, and we'll talk about those next time. This diagram shows you the differences between acute and persistent infections. So on the top is an acute infection, and what we're looking at here is virus production and disease on the y-axis with time on the x-axis. So the blue lines represent virus production, and the red bars are episodes of disease associated with infection. So on the top is a typical acute infection caused by rhinoviruses, rotaviruses, influenza viruses. You have disease associated with virus production, and then the virus is gone. It's cleared, and you recover, and that's the end of the infection. So that's what acute means. And the rest are different kinds of persistent infections, which we'll talk about next time, and they simply involve, again, infections that last the life of the host. There can be bursts of virus production, intermittent bursts, or there can be continuous virus production. This happens to be a persistent asymptomatic infection where there's no disease. Or on the bottom, you can see a persistent infection where the disease itself results in death. And we'll get to those next time. But for today, acute infections involve a rapid onset of infection or reproduction, a short disease course, and it can be mild or it can be severe. Whether it's acute or not has nothing to do with the severity. Large numbers of virus particles are made, and eventually the infection's cleared by the immune response, and you have some kind of immune memory as a consequence. Here's the course of a typical acute infection. Again, we're looking at virus growth on the y-axis, time after infection on the x-axis. And so at the very left, we have entry of virus into the host. The virus begins to replicate. Virus titer is shown by the jaggy red line. And then that, of course, that initial replication engages innate defenses, the production of interferons and other cytokines. Most infections are controlled by the innate response, but if it can't be controlled and the virus continues to replicate, it then exceeds a threshold. Beyond that threshold, the adaptive responses are engaged because the innate responses weren't sufficient to clear infection. And so then the virus replication eventually peaks. You have adaptive responses associated with clearance of the virus, and then, of course, immune memory. So again, it's an acute infection, a very defined period, very different from persistent infections, which we'll talk about and why they occur. A characteristic of any infection, not just acute infections, but persistent infections as well, is there is what we call an incubation period. An incubation period has a very specific definition. It is the initial period before symptoms of the disease are obvious. And not signs, but symptoms. You'll see why in a moment. During the incubation period, which is diagrammed on this illustration at the top, virus genomes are replicating. So you've been infected, the virus is replicating. The innate responses are engaged. And depending on the virus, you may actually shed virus and transmit it during this period. Some viruses are transmissible during the incubation period and others are not until you have clear symptoms of infection. So this picture suggests that you were infectious only in a period beyond the incubation period. And that's not quite correct because, as I just said, some viruses are infectious during the incubation period. Now, during that period, there are signs of infection for sure. You just can't tell because by definition a sign is something that you can't sense. You can't sense that virus is replicating in you unless you have symptoms caused by interferon, say. But there is a period of time before innate responses are engaged where you don't have the fever and malaise and so forth caused by the production of interferon. So that's the incubation period. And again, you may or may not be infectious during the incubation period. It depends on the virus. So that's why I say the initial period before symptoms are obvious, because clearly, if I took some of your blood, I could find virus in it, and that would be a sign of infection, something, again, which can be measured but which you cannot feel. Incubation periods vary greatly among viruses, from 1 to 2 days here for influenza virus to between 50 and 150 days for papillomaviruses that cause warts. If you ever had a wart on your hand or the bottom of your foot, that's called a papilloma. And that's caused by papillomaviruses, of which there are many. And a few of those are associated with sexual transmission. Some of those are associated with cancers. We'll talk about that when we talk about vaccines, because we do have a vaccine to prevent their transmission and to prevent cancers. These viruses have a very long incubation period. And you can see viruses have intermediate incubation periods as well. Couple of days, a week, 2 weeks, 3 weeks, 4 weeks. Viruses with a short incubation period. In general, that's because the virus replicates at the same place where it produces symptoms. So rhinoviruses come into your upper tract, they replicate there, and that's where you get the symptoms of a common cold. So it doesn't take a lot of time for the virus to do that. Viruses with a longer incubation period tend to have to spread from the initial site of infection. Not always, though, there's always an exception in biology, but for the most part, these viruses have to spread. For example, poliovirus enters the intestinal tract and most of the time stays there, as you will see, but when it has to get into the CNS, I shouldn't say it has to get in, when it enters the CNS, that takes longer time because the virus has to spread from the gut to the brain or spinal cord. The other term here, which you may encounter, is called prodrome. That's the period before we see symptoms characteristic of the disease. So if you had measles, the measles rash is characteristic of measles. So the prodrome is the period up until you see that rash. So it's beyond the incubation period for most viruses. That's the prodrome. Many acute infections are inapparent, and we mentioned this a long time ago. That is, you are infected, the virus is replicating, but there are no symptoms. That is, again, you can't tell you're infected. So you go about your work and you infect other people. But of course, an inapparent infection can go beyond the incubation period to where there's lots of virus in you, but you still don't have symptoms and you can spread it to other people. So you make enough viruses so that they can spread in the population. How do we know this? How do we know there are inapparent infections? Anybody know? Could you harbor a guess? I've just told you there are inapparent infections. Why should you believe me? Could be fake news. What is a way to detect if someone's been infected? A regular blood test. What kind of test? A blood test. Yeah. Take some blood and look for antibodies, right? So we do serological surveys of populations, and we can tell, and when you take blood from someone, you can ask them, have you ever had influenza or this whatever other disease? And you can tell that a lot of people are infected and don't show symptoms. Polio is an obvious one because if the person has not been paralyzed, you can believe them. So that's how we know that there are inapparent acute infections. And I call these well-adapted pathogens. These are viruses that have been in us, in humans, Homo sapiens and our ancestors for many, many years, and they have adapted to replicate in us with causing minimal disease. You'll see when we talk about evolution and emergence, every virus that we have today, we got from an animal of some sort, a non-human animal. Either Homo sapiens or our ancestors acquired it from an animal. The most recent ones tend to be more virulent because they haven't undergone this long adaptation in the host. And for example, HIV, we think jumped into humans from a monkey probably around 1920. So that's a pretty new one. Ebola, we think it jumped into humans in the 70s, although that doesn't spread among people. So every outbreak is a new jump. So these viruses that cause inapparent infections tend to be well adapted. And there's a number. Polio, over 90% of polio infections are inapparent. And that's a problem because, as I've said before, I believe here, in the heyday of paralytic polio here in New York City, the practice was go from house to house. The police would go from house to house, look for paralyzed kids, and quarantine them in the hospital. Quarantine, by the way, means originally 40 days. That's what the Latin means, to put someone somewhere for 40 days. Now it just means to isolate someone until their illness is over. And that did nothing for polio because paralysis is 1% of all infections. And 90% of the people have no symptoms and they're walking around spreading it. So quarantine was useless. And in fact, for many viruses, it's useless. This issue between transmission and the incubation period is very interesting. It has practical implications. I don't know if you remember, a number of years ago, there was an Ebola outbreak in West Africa, right? A doc from Colombia went there to work. He came back and he was at home. He went bowling in Brooklyn, and then the next day he developed a fever, and immediately he went to Bellevue, and they saw that he was infected. He was an at-risk person because he'd been working in Africa. There was almost a lawsuit that he shouldn't have been going out and bowling on the night before because he would be infectious. And lawyers actually called me to get my opinion on this, and I told them, for Ebola, you do not spread infection unless you have symptoms. That's one of the viruses we know, and we know this by just looking at all the past infections as well. But that was and other cases as well. You know, there were cases of nurses in Connecticut who went bike riding after they came back, and people were all upset about this, and they didn't have any symptoms. So this can have practical consequences. No suits were ever brought to bear. Acute infections tend to be common health problems. Not that persistent infections are not, but these have a particular issue. The viruses we're going to talk about, like influenza virus and norovirus and others, they cause a lot of infections every year, and they're hard to control because by the time you feel sick, you have already shed virus and passed the infection to others. Which of the following do acute infections and incubation periods have in common? The virus is not replicating, no symptoms are visible, immune defenses are engaged, the immune system does not respond, or all of the above. The answer is C. What I'd like to do today is to pick a number of acute infections and go over them as ways of illustrating some of the principles. So these are 5 viruses that cause acute infections. And I pick them because they're transmitted in different ways. You know, influenza virus and measles virus are respiratory transmitted infections, but they're different enough so it's worth doing both. Poliovirus and norovirus are transmitted by fecal contamination, ingestion of fecally contaminated material. And again, they're different enough worth considering. And West Nile virus, how is that transmitted? Mosquitoes, exactly. So that's a different one to look at. Influenza, we are almost at the end of the flu season. April, May is the end. This has been a rough flu season for a variety of reasons. Influenza, an envelope virus, of course. With the segmented RNA genome, there are 3 types. A and B, which are part of the vaccine. They cause similar disease. And C, mostly inapparent or asymptomatic infections, so we don't worry about C. And A is unique because it causes pandemic. A pandemic is an epidemic that involves the entire planet. And a hallmark of these viruses is antigenic variation. It evades from year to year the vaccines, and that necessitates reformulating the vaccine almost every year. And then every 20 or 30 years, there's a major change in the antigenic structure of the virus, and that causes a pandemic because no one has any immunity to that. These viruses are transmitted by the droplets made by coughing and talking. Sneezing, I told you a while ago, by one study doesn't seem to make any difference. It's still on this slide for historic reasons. And remember, there are droplets of different sizes. So those small, whenever you sneeze, talk, even or breathe, you're making droplets of all sizes, and the smallest ones travel the furthest. Direct contact with infected people, if you have a runny nose and you touch the person, you can contaminate your fingers, or surfaces as well. The virus can remain infectious for a while on certain surfaces. And I'm thinking of a study done in Switzerland where they pipetted influenza virus onto banknotes. Who else would do this but the Swiss, the banknotes, right? They have so many of them. And they asked, how long does the virus retain infectivity on a banknote? The idea being, you go to the bank. Those notes are pretty filthy. You think about it, all the people who touch them. The sooner we get away from them, the better. Anyway, the virus can remain infectious on up to 2 weeks. And if you have mucus with the virus, it lasts even longer. So the mucus stabilizes the infection. Beware of the banknotes. I always tell people, when you touch public surfaces, anything, a subway pole, a banknote, just don't put your fingers in your eyes or your mouth until you wash them. If you can do that, you will definitely avoid getting infection. I have a colleague at Columbia who won't even shake your hand. He does this nonsense with the elbow, And he says he doesn't get colds anymore, but I don't believe him. The virus enters your respiratory tract, replicates in the upper tract, the nasopharynx, the epithelial cells that are lining the respiratory tract, and influenza can spread all the way down to the bronchioles and the alveoli. So not only replicates in the upper tract, it spreads through the trachea, and in fact, much of the infection is focused in the trachea. So if you get flu, you get a lot of substernal pain because CTLs are in there killing your lung cells. It really wipes out the epithelium. And if it gets into your alveoli, that's really bad, because then you can get viral pneumonia. Your alveoli will fill with fluids, and you can't breathe very well. We've also recently found that the virus can replicate in your eye. So some strains are able to replicate in the conjunctiva and the cornea, the epithelial cells there. The virus will drain. There's this lovely duct that goes from your eye into your nose. The virus will drain there into your nose, and you will inhale it, and it'll go into your lungs. And these numbers here represent the different kinds of sialic acid receptors that are present in your upper tract, in the rest of your tract as well. And if you remember from a long time ago, the alpha-2,3 linked sialic acids are preferred by avian influenza viruses. So a lot of people who work with birds, chicken farmers, they get eye infections. So you can get conjunctivitis, and then it spreads to your lungs. The human strains typically prefer alpha-2,6 sialic acids, and you can see there are lots of them in the upper tract as well as the lower tract, and there are some up here in the nasolacrimal duct, not many on the eye though. So an eye is just another portal of infection. Keep your fingers out of your eye. Uncomplicated influenza, short incubation period, quick onset. You remember exactly when you started to get sick. You get high fevers, muscle aches, malaise, anorexia. A couple of days, the fever peaks, then it goes away. And then, if you remember this, after the fever starts to decline, then you get really bad respiratory cough. And it's productive. You make a lot of mucus after the fever is gone. And you can cough for a week or two, lasts a long time. You can feel really lousy because it wipes out your respiratory epithelium, and it's got to regenerate. Way different from a common cold where you can recover quickly. There are two ways you can diagnose flu. If it's the flu season, which is November, October to April, May, and you have influenza-like illness, which is fever at 100 degrees, cough, or sore throat, that's it. You go to a doc, they will say, you have flu, it's the season, you have the right symptoms. There are lab tests, which a doctor can do in office, but they don't have very good accuracy. PCR, viral culture, and serology are very accurate, but they take too much time, and you will not be able to take an antiviral. Remember, you have to take an antiviral within a day or two of the onset of symptoms. Otherwise, it's not worth taking. It's a waste of money. Diagnosis is easy in season because if they suspect you have flu, they won't even bother with a test and just give you antiviral. Here's some growth curves to give you an example of the course of infection. These are actually volunteers who were given mild strains of influenza. So this is one of the viruses that you can actually infect people with experimentally. So we inoculate people on day 0. You pipette a little virus into the nose. And then we measure viral titers in mucus or exudate. And then this has also symptom score. They ask you how you feel, and they measure one of the things they do is have you blow your nose and they measure the weight of the tissues, see how much they weigh. The more mucus you make, the more the tissues weigh. So here we have viral shedding. You can see it goes right up after inoculation. It peaks at day 2 or 3, and the symptoms are not far behind, and then they decline pretty rapidly. But in here you have the cough that persists for a long time. On the right is another similar study. You inoculate people. We're looking at virus. Here, the first curve, and this is interferon, that dotted curve underneath it. So you can see interferon goes up very rapidly and declines. And then these are antibodies in the nasal wash, in the serum. So you can see they come up way too late to be of any use for this infection. But they're good to make memory so that the next time you can respond. Here are a series of slides of data. Flu is one of the most studied infections in terms of the data we collect, because global laboratories coordinated by WHO, all over, thousands of laboratories that collect samples to see what the current circulating strains are, so we know how to make the vaccine. So this is a summary of outbreaks over a couple of year periods, 2004, 5, 6, and 7, just to show you the seasonality. This is from the North American laboratories, and it's peaking in the winter months, as you can see here. Nothing in the spring, summer, or fall. Peak every winter months. And the colors tell you which different kinds of influenza viruses they mainly get. And the predominant ones are the H3N2 and the H1N1. Those are subtypes defined by the hemagglutinin and the neuraminidase glycoproteins. Those are in red and blue. You can see some years the red, the H3N2 predominate, other years the blue. And then influenza B viruses is a smaller fraction. So that's to show you the seasonality. This is this year, these are the numbers this year. And again, number of positive specimens. So here are thousands of specimens that are collected. So again, there are regional laboratories that do this and they send the results to CDC. This is the week of the year. So that's the 40th week of 2017. It's getting to the end of the year, obviously. There's week 52. So that's December. So you can see it's quite high already. And you can see that the predominant virus this year is an H3N2 virus. Very little H1N1 in the brown. And we've already passed the peak. So this is now week 12, January, February, March, right? 4, 8, 12, right? So it's really over, essentially. There's not very much flu, but it peaked in December and January. On the right is what we call pneumonia and influenza mortality. This is from a data surveillance system where they look for this. So they're combined because flu often leads to death in the elderly and the very young by pneumonia. So they combine flu and pneumonia mortality because when you die often they don't put influenza on your death certificate, they'll put pneumonia, but it's often secondary to influenza. And these are the numbers for different years, 2013 to the present. And the big red spikes are the percent of deaths due to pneumonia and influenza. And you can see some years there's a big spike and it exceeds what is predicted. The black lines are what we would predict to be the normal number of deaths. And you can see in some years the flu isn't so serious. Some years it's very serious. And this year we had a big spike of pneumonia and influenza. And so this was a pretty serious year. The H3N2 virus tends to be more virulent than the H1N1. This year H3N2 is predominating, and that probably in part explains this. The other part that explains it is most people don't get the flu vaccine, less than 50%. And this year there was a mismatch between the H3N2 and the vaccine. So all of these things combined to make it a particularly difficult year. This is an interesting statistic, and there are tons of these graphs. I'm just showing you a few of them. Percentage of visits for influenza-like illness reported. This is many different years. So there's the 2009 pandemic year when a brand new strain emerged and nobody was immune. So we had a big jump in the number of hospital visits, outpatient visits for influenza. And then you can see in other years it wasn't as high. It's the blue one, 2016, 17. This year the red, as you can see, very serious. So not only mortality, but a lot of people going for medical care. Typically, the virus in the US causes about 35 to 50 million cases of influenza every season. And the deaths can go from 3,000 to 50,000 is the prediction for this year. This is a range of the past 31 years. It's not always high. It can be low, or it can be towards 50,000. And that's why we have a vaccine, because it can kill you. It could kill any one of you. You are healthy, you could get flu and die if you're not vaccinated. So you really should get vaccinated. There's no reason not to. Just because you're 22 and you've never had flu doesn't mean you won't get it next year. So you should get the vaccines. There are also complications of influenza. You can get pneumonia, as I've said. You can get bacterial pneumonia, secondary. You start to recover from the viral infection, and then all of a sudden you get a bacterial pneumonia, generalized muscle pain. Your heart can get involved because your lungs are overworking. It puts a stress on the heart. Many people get heart attacks with influenza, especially elderly people or pregnant women. And then there is a syndrome called Reye's syndrome, which is an encephalopathy associated with liver damage. There are interventions available. You can go and buy stuff at the pharmacy that says this will make you feel better. That's what I mean by non-pharmaceutical. Or there are antiviral drugs, Tamiflu, Relenza, and Flumidine. Tamiflu is taken orally. Relenza is inhaled. That is an inhaler. And they're both similar inhibitors of neuraminidase. We'll talk about those later. And then there's another antiviral, Flumidine, which is not used much because every virus out there is resistant to it. And then, of course, there's a vaccine which would prevent you needing to have any of these. So you should take the vaccine, even though it's not perfect. Someday it will be really good, but today it's not. I'm just curious, how many of you got a flu vaccine this year? Very good. I think it's higher than usual. All right, next question. Which of the following is characteristic of uncomplicated influenza? Transmission by respiratory droplets, incubation period 1 to 5 days, fever peaks within 24 hours, coughing and weakness can last for 2 weeks, or all of the above. Okay, most of you got the right answer, all of the above. Poliovirus, causative agent of poliomyelitis. This is an icosahedral particle, no envelope, plus stranded RNA. And this virus is ingested along with fecally contaminated material. So you ingest the virus, you put fingers in your mouth that are fecally contaminated, or you eat food that's contaminated. The virus goes into your intestinal tract, passes through the stomach, low pH digestive enzymes, and it gets into your small intestine where it's high pH and bile salts and proteases, and the virus still retains infectivity. Remarkable. Replicates in the intestinal cells, the mucosal surfaces, gets into the subepithelial tissues, gets into the blood via the lymph node, and can spread to the spinal cord and brain. But this is a rare event. Most of the time the virus is replicating in the intestine. It's shed and spreads to other people. And it's a really efficient virus in terms of spreading without any disease. But about 1% of infections, the virus enters the neuromuscular junction. The virus enters the nerve, goes up to the spinal cord, and begins to replicate in neurons and destroying them. You destroy enough neurons that innervate a muscle, the muscle becomes paralyzed, and that's what poliomyelitis is. But it's an accident. This is a section of one of our transgenic mice expressing the polio receptor, infected with polio and paralyzed. It is stained with RNA to show where the virus RNA is. So these are the green dots. These are all neurons that are infected. And these are immune cells infiltrating into the cord. So eventually enough of the neurons are destroyed, and that causes poliomyelitis. Here's a blow up of a single neuron. You can see it's full. The axons and dendrites are full of virus. This is a figure from a 1950s textbook of medicine. If you look at a textbook of medicine today, you won't find any more on polio. They don't have it there because there's no more polio in the US. This is a really interesting figure. First of all, it's hand-drawn, which I think is quaint, but that's how we used to do things. Here we have course of infection, starting with exposure, and on the top is temperature, and then we have virus in different places, and then we have antibody, and also this is virus in the CNS in the bottom, this is virus in the lymph and the blood, and this is virus in the alimentary tract. So you can see after about a 7-day incubation period, there's a spike of temperature associated with headache, sore throat, nausea. Most people get better after that and never get polio. So they would never think they had polio. Some people, the virus continues to replicate, and there's a second spike in temperature associated with more headache, nausea, stiffness, pain, and paralysis. So there the virus is getting into the central nervous system. And you can see you are shedding virus in your saliva and in your feces. Look at feces for up to 30 days here and longer. There's virus in the blood. As you can see, that correlates with that first spike of temperature. And then the bottom shows virus in the CNS. So again, that correlates with paralysis. And again, I told you this is about 1% of infections that the virus gets into the CNS. So this is an exclusively human virus. There are no animal reservoirs, and that's one of the reasons why we can eradicate polio. It's almost eradicated. This is seasonal. It peaks during warm months. We think because people, kids especially, have contact with each other during that time. There is a complication called post-polio. 30 to 40 years after you have polio, most people can have their limb functions returned by therapy. They can walk again. A number of these, about a quarter to 40% of these individuals, start getting paralyzed again. They have to go back to a wheelchair. This is not an infectious process. The virus is gone. What it is as you age, you naturally lose neurons, but normally you would have lots of backups, but if you've had polio, you don't, because a number of them have been destroyed. And so you get paralyzed at an older age and it can be very serious. We still have people undergoing this because the polio epidemics were not that long ago in the US. These are cases of polio in the US from 1950 to 2007. And you can see it peaked in the 50s. We first became aware of polio at the turn of the 20th century. We knew very little of it before that. And then all of a sudden there were big outbreaks from the beginning of the 20th century. And there were thousands of people involved. And an activated vaccine was introduced in 1955, and then an infectious oral vaccine in 1962. And we had the last case of polio in 1979 in the US. It's no longer present in the Western Hemisphere. There are only 3 countries, Nigeria, Pakistan, Afghanistan, where we still have ongoing polio, so it's almost been eradicated. We'll come back to this later when we talk about vaccines. Measles virus, another acute infection. It's a different kind of virus. It's an envelope virus with a helical nucleocapsid. It's a lovely EM of the helical nucleocapsid, which is long and all wrapped up in the envelope. Paramyxovirus. This is one of the most contagious human viruses. The R0 is 15. The R0 is the average number of people you will infect if you are infected with a virus. So some viruses are 1 or 2. Measles is a big one. It's 15. I used to say in the old days, before vaccination, if I had measles and I wasn't symptomatic and I didn't know it, I would be exhaling it, and most of you would be infected, including you all the way in the back, because my droplets would reach you. It's really remarkably infectious. So there's one serotype, and it doesn't undergo antigenic variation. So you have lifelong protection after infection. It's a respiratory transmitted infection. You can shed virus before the rash appears, so you can shed before you know that you are infected. And this one is interesting because there are very few asymptomatic infections. Almost everyone that gets infected with measles has a rash. And in the days before the vaccine, you would go to school with a rash, they'd send you home, or hopefully your parents would keep you at home so you wouldn't spread it to others. The way the virus replicates and spreads in you, you inhale respiratory droplets from another person who's infected. It replicates not in the epithelium, makes its way to the lung, and actually replicates in immune cells that are patrolling the epithelium. The immune cells then get infected. They bring the virus in, and it spreads into your blood, establishes a viremia. The virus is then spread to multiple organs, including the skin, leading to the characteristic rash. In some cases, the virus can get in the brain and cause encephalitis. About 1 in 1,000 measles infections lead to encephalitis, so this can be a serious infection. In order to spread the virus to another person, what we believe happens is the virus is in your blood, makes its way to the basolateral surface of the respiratory epithelium. It's brought there probably by infected immune cells. There are receptors for the virus on the basolateral epithelium. They are not present on the apical domain of these cells. And that's why to infect you, the virus can't infect the epithelium. It has to infect immune cells that are scattered throughout the epithelium. But on the way out, The virus attaches to receptors on the basolateral surface, replicates in these cells, and then is spread. And then as you talk and sneeze and cough, you spread it to other people. Really unusual mechanism. Probably selected for because it needs to get out by the way it came in. So that's a child with measles all over the body. Also early in infection, you can see spots in the mouth, these little white things. You can tell that a kid has measles before the rash because these are they're called Koplik spots. They're almost diagnostic for the infection. What they are syncytia, lots of infected cells that are fusing with many nuclei, and they have this white appearance. High fever, runny nose, cough, conjunctivitis, and this rash from the face to the extremities. But this has complications that are serious. Encephalitis, 1 in 1,000 kids, and they can have serious neurological consequences as a result. Bronchitis, pneumonia, ear infections. So the fatality rate is 1 in 2 per 1,000 infections. That's pretty high. But in countries with poor nutrition, as I mentioned last time, it's much higher, 28% fatality rate in countries where kids are not properly nourished. Another complication is SSPE, subacute sclerosing panencephalitis. This is a chronic degenerative neurological disease where 10, 20 years after getting measles, suddenly the child begins to undergo neurological deterioration and eventually will die. And it's because measles remained in the brain and has been replicating there. And as I said, this virus is a great immunosuppressor. And this causes a lot of secondary infections, which turn out to be the main cause of death in third world children. We made a vaccine because we used to have 3 to 4 million cases of measles a year in the US, 400 to 500 deaths, a lot of hospitalizations, chronic disabilities from encephalitis. So the vaccine has stopped endemic transmission. It is given as what we call the MMR vaccine, measles, mumps, and rubella, 3 viruses together. It's an infectious vaccine that is injected. Unfortunately, in 1998, Andrew Wakefield published a paper saying that this vaccine caused autism. Paper was wrong. It was retracted by everyone except him. But unfortunately, a lot of parents have continued to believe that he's a demigod and he's right and battling the evil medical establishment. And many parents will not immunize their kids against measles because of this fraudulent paper. And Andrew Wakefield, by the way, has since been debarred of his medical license in the UK. He moved to Texas, where they welcomed him. That's why they have a town called Waco in Texas. Is anyone from Texas? I'm sorry, I don't mean to insult you. Get immunized. Since he moved to Texas, the number of children whose parents refuse to immunize them has gone up 1,000%. And there is zero evidence that this vaccine causes autism. It's been subjected to so many clinical trials, it's unbelievable. Yet he continues to hold that this does cause autism, and we are the victims of evil pharmaceutical companies. It is just not true. I'm sure you don't believe it, but some people are very swayed. Because of decreased immunization, and this occurs in pockets, parents get together, are you going to immunize your kid against? No, I'm not going to. And then they all don't, and you have pockets of susceptibles, and you have outbreaks in the UK and Ireland initially, because that's where the paper was published, and then these people spread the virus to the US, so there have been lots of imported outbreaks as well. Here is the epi curve for measles in the US, starting from 1950, lots and lots of cases. Vaccine license, 1960s, boom. Really big drop in cases of measles. In the inset, these are deaths per year, so we went down to very few deaths. But look, there's still measles, and there are little outbreaks now and then. And that's because periodically people do not vaccinate their kids. Here are the details by year in the US, 2010 to the present. You can see every now and then there are outbreaks of 100 or 200 cases. Big one in 2014, 700 cases. I think this may have been the one associated with Disneyland in California. Lots of parents hadn't immunized their kids. They all congregate at Disneyland. It's a big pool of susceptibles. All you have to do is introduce virus and you get an outbreak. So you can see by this state map of the cases, the dark colors are a lot of cases, so they tend to cluster. And they tend to cluster in places where people get together and congregate and decide as a group that they're not going to get the measles vaccine. There are lots of states without cases, and good for them. We have 1 to 4 here in New York and northeastern area, but California is a guilty one. This is 2015. So again, there's no excuse for this. This is a completely vaccine-preventable disease, and the vaccine is harmless. It does nothing to you. You want to have your kid get encephalitis, it's a lot worse than anything the vaccine will do. These are global cases in a 6-month period, and you can see the colors tell you how many cases. The dark red is the worst, over 1,000 cases in that period. As you can see, there's lots of measles globally. You know, we're between 10 and 99 every year. And it's because a lot of these countries don't immunize. They don't have the vaccine in order to do it. We could have 0 measles. We could actually eradicate measles because there's no animal reservoir and you have lifelong immunity. Those are the 2 requirements for eradication. WHO wants to eradicate measles at some point. But if we can't distribute the vaccine, look, even in Europe, there's some places. This is one of the best buys in public health from 2000 to 2014. 17 million deaths were prevented by immunization. How can you argue with that? Andrew Wakefield never brings up these numbers, by the way. He always says that the vaccine is evil. Which of the following is a good reason to get measles vaccine? There is a 1 in 1,000 chance of acute post-infection encephalitis. There's a 1 to 2 in 1,000 chance of death. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections or all of the above. 100%. Right. So for the 500th episode of This Week in Virology, we are going to Austin, Texas, to the University of Texas. And one of our guests is going to be a woman named Jenny Sue, who was a Columbia Law graduate. She moved there, and she started an organization called Immunize Texas. So she's going to be one of our guests and tell us about why she did this and how it's going. So I'm really glad there are grassroots people like that want to do the opposite of Andrew Wakefield. All right, another kind of GI-transmitted virus. Here's a neat figure. About 200 million people have gastroenteritis every day. And the amount of water they pass equals how much goes over Victoria Falls in 1 minute. 65 million liters per minute. It's coming out of people who have gastroenteritis. So gastroenteritis is diarrhea and vomiting. And one of the viruses that does this is Norovirus, which is a small RNA-containing virus with a lovely capsid. Here on the right, you can see the icosahedral symmetry. It has a plus-stranded RNA, and it causes about half of all foodborne outbreaks of gastroenteritis every year in the U.S., 23 million cases a year. There are other causes as well, but this is a big one, and you'll see why. So these are the known causes of foodborne illness in the U.S. from 2006 to 2010, norovirus, 49%, bacteria, 40%, and other causes. You know, before noroviruses were discovered, we thought all gastroenteritis was bacterial. And now we have noroviruses and rotaviruses, other causes of gastroenteritis. It's really changed our impression of these kinds of diseases. Now, this virus causes what I call a 2-bucket disease. 1, 2, because you need a bucket to vomit in and a bucket for your diarrhea. And I learned that from a microbiologist. Someone told me today there are 3-bucket diseases, but I can't imagine what they would be. This virus is spread by fecal-oral contamination. You ingest material that has norovirus on it, passes through your stomach, remains infectious like poliovirus, and in your small intestine it causes blunting of the villi. So here at the bottom are 2 photographs of villi, on the left from a normal small intestine, on the right from a person infected with norovirus. And you can infect people with norovirus. A lot of research up until recently was done by infecting people because it's not terribly serious if you don't mind 2 days of vomiting and diarrhea. At Washington University in St. Louis, for example, they hire medical students. They pay them $300. On Friday, you drink a little filtered stool from a person with diarrhea because you can't grow it in cell culture. And they add some flavoring so it tastes good. And then you get sick over the weekend and they take samples from you and they can do their study. And then Monday you can go back to class because you're fine. And for that you get $300. So this is a virus where for years you could not grow it. So you had to infect people to get the virus. Now again, it goes into your intestinal tract, replicates. And we don't know why the virus causes these symptoms. I told you last time for rotaviruses, they make a NSP4 toxin protein that causes diarrhea. We don't know why noroviruses do this. So for years, people, if they wanted to grow norovirus, they would find a volunteer, pay them, and you would ingest a stool extract, a stool filtrate. For years, people haven't been able to grow the virus in cell culture because that's what you need to do to figure out how it works. And a number of years ago, people found that not only does the virus grow in B cells, that's interesting, not the epithelium of the gut, but in B cells, which are below the epithelium in the subepithelial spaces, but you need bacteria for the virus to grow in these cells. So here's an experiment where they take some diarrhea from an infected person. They either filter it or don't filter it. They filter it to remove the bacteria, right? 2.2 micron filter, the virus goes through, the bacteria remain on the filter. Unfiltered, it's growing. This is viral genomes in time post-infection. If you filter it, it doesn't grow well. So you need enteric bacteria for the virus to grow, because it turns out the virus binds to bacterial proteins, and that's how it gets taken into the subepithelial spaces. This was an accident. Someone forgot to filter the material one day, and they found that it grew in cells. You don't need bacteria to feed it to someone and get infection because they have their own gut bacteria. But they were trying to figure out how to get it to grow on cells, and they had forgotten to filter it one day, and it grew. Serendipity. Here's the model for how these viruses infect your intestine. This is the intestinal epithelium. And in your gut, of course, you have tons and tons of commensal bacteria. And the virus, which is shown in a little red star, binds to components, carbohydrates in particular, that are present on the bacteria. The bacteria then get transcytosed across the M cells. Remember, the M cells are always sampling the contents of the gut lumen. Bacteria get pulled in with virus, and then the virus replicates in the B cells that are present below in the subepithelial space. How that leads to diarrhea and vomiting, we don't know. But now we do know how to grow it in B cells, which is good, so you can make a vaccine. You don't need many viruses to get you sick. 18 particles in food or hands can make you sick. So the number of particles that fit on the head of a pin could infect 1,000 people. So the way it works is fecally, orally spread, but there's some evidence that if you vomit, and you do vomit after this infection, you make an aerosol that has virus in it, and people can inhale that and swallow the virus, and it will infect you. Fomites, food, water, environmental contamination, oysters and raspberries. Oysters, of course, because they're filter feeders, and they'll take up a material from contaminated water. If you wash raspberries with contaminated water, you can contaminate it as well. And it's often transmitted by food handlers who are asymptomatically infected. They're shedding virus. They don't have good hand hygiene. This is the virus that resulted in all those signs in the bathrooms of restaurants. Employees must wash their hands before returning to work. That's norovirus, because we figured out that those people were going back with dirty hands and getting the virus into food. Incubation period, sudden onset of vomiting, 28 to 60 hour, pretty quick illness, and here 30% asymptomatic, and that's how it mainly is spread in these institutional settings like cafeterias and cruise ships. Peak of shedding 1 to 3 days, but you can shed for 56 days after infection, after your illness. So you can imagine people get better and they go back to work, and if they're a food handler, boom, that's how they spread it. There's some issue with immunity here. You don't get immune after infection. It's only short-term immunity. You can be reinfected multiple times in your life. So how that will translate into a vaccine is difficult to parse. Only humans seem to be a reservoir, but there's some evidence that caliciviruses in marine mammals can infect people as well. There's a peak in cold weather. And here the outbreaks are often in semi-closed environments. And these, of course, have been increasing in recent years, the last 50 years or so. And so the incidence of this has increased, nursing homes, hospital, cruise ships, all these kinds of environments where lots of people are together, and that's a good way of spreading these infections. At the moment, we have no way to treat norovirus infection except giving people fluids, which usually is enough. As I said, it's not a very serious illness. And they are making a vaccine. These are how the viruses are transmitted. On the left are the settings. You can see that the biggest is restaurants, catered meals, 40%, retirement centers, nursing homes, hospitals, schools, daycare centers. I think there was just an outbreak in the Olympics, right? people got gastroenteritis. It was probably this. You put all these people together, an infected food handler, boom. Cruise ships is not a major form, or not a major venue, but it gets a lot of attention. And on the right are the modes of transmission. Person to person, 88% is the biggest, and foodborne is only 10%, interestingly. Here's an interesting article I found a while ago about an outbreak of norovirus on airplanes. And this particular incident, a sick passenger vomited into the bag, but that aerosolized the virus, and a lot of other people got infected on the plane, and the flight attendants also got infected, and they went on another flight and spread it to those individuals as well. So this virus can go by aerosol as well. And this, the cruise ship gastrointestinal bug, this is a virus, it's not a bug. So if you ever write articles, please don't call viruses bugs. So cruise ships get a lot of publicity when there's an outbreak of norovirus, because the whole ship has to come back and everybody has to get off and they have to decontaminate it. And the reason usually is a food handler who is infected asymptomatically. And here are just some recent cruise ships where we know that noroviruses have caused some of the outbreaks, but there's some where we don't know what the agent is. Could be there are other viruses out there as well. This is Disney's name for their cruises, the happiest place on earth. I wouldn't go on a cruise ship if you paid me $1 million. Don't touch them. Why are they associated with cruise ships? Because we track the illness. We're really attuned to it so that we can, because this is an expensive ordeal. Close living quarters probably helps to spread, and then you're always bringing new passengers in, so probably that assists as well. But as I showed you, it's not a major way of spreading the infection. These are ways you can protect yourself. Wash your hands, rinse your food really well, and don't eat the mince when you go out of the restaurant. They're really fecally contaminated. People have done studies of these things, and they're covered with fecal coliforms because people come out of the bathroom and they take one on the way out of the restaurant. Even if they're wrapped in cellophane, don't take them because the cellophane is contaminated and it will contaminate your fingers as well. Okay, remember that. If there's one thing you remember, don't eat the mince. Our last virus is West Nile. One of my favorite cartoons. We're pretty sure it's West Nile virus. West Nile is not in Egypt, actually. It's in Uganda. So this cartoonist didn't take a virology course. The West Nile district of Uganda is where West Nile virus was first identified. It's nothing to do with Egypt. Isolated in 1937. And we actually did not have it here in the Western Hemisphere until 1999 when it came to New York City. As I told you before, came from probably Israel because the isolate in New York was identical to an isolate from an Israeli goose on a goose farm. It infects many kinds of birds, many kinds of mosquitoes, and other vertebrates. So it has a very broad host range, and that's why once it entered the U.S., it spread so quickly. Infects a lot of different mosquitoes and birds and other animals, and not just humans. If it just infected humans and mosquitoes, it might not have spread. But now it's all throughout the Western Hemisphere. West Nile is in green. And basically, wherever culisine mosquitoes are, you can find West Nile virus. So this is a flavivirus, of course. It's an envelope virus with a plus-stranded RNA genome, like Zika virus and dengue virus. The primary transmission cycle of this virus is among birds transmitted by mosquitoes. This is the natural cycle of the virus. You can go out and you can find West Nile in many different kinds of birds. When it came to the U.S., the crows in the U.S. had never seen it before, and so they died, and many other birds died as well. One of the sentinels was the Bronx Zoo. A lot of the exotic birds in the Bronx Zoo were dying, and eventually it was because of West Nile. Hard to prevent birds from being bitten by mosquitoes. So the virus is spread among birds in the wilds by mosquitoes, and then we and horses, by the way, are incidental hosts. You know, we get bitten by the mosquitoes that have taken a blood meal on an infected bird, and we can get infected. Now remember, that mosquito has to take its second blood meal, and less than 1% of mosquitoes do that. Horses can get very sick from West Nile virus infection, and if you have a racehorse that is worth millions of dollars, you immunize it. There was recently a West Nile vaccine developed just for horses, and it's to protect your investment. These are the mosquitoes, the culicine mosquitoes, which are prevalent in many parts of the US. Short incubation period, and then 20 to 30% of people develop a flu-like illness, which is called West Nile fever. Flu-like, you can imagine what the symptoms are: fever, headache, malaise, etc. Can't be distinguished from other infections, and highly asymptomatic. A number of people, 1 in 150, get neuroinvasive disease. The virus enters the central nervous system. You get headache, you have eye problems, muscle weakness, you can have cognitive impairments. So these people can have long-term sequelae as a consequence of this, even polio-like flaccid paralysis. And there's 10% mortality associated with this, in particular in elderly people. So a vaccine is definitely needed for this. Either that or monitor your mosquito exposure. Try to avoid them. Just don't go outside. Here's the pathogenesis of West Nile virus infection. Again, there's a natural cycle among birds via mosquito, but when it bites a human, it puts virus into the tissues surrounding blood vessels. Remember, mosquito doesn't actually inject virus into your blood. When it finds a blood vessel, it sucks blood out of it. But when it's probing for a blood vessel, the proboscis of the mosquito is releasing anticoagulants and compounds that prevent you from feeling the mosquito. And when it releases those, viruses coming out of it as well. So then in the subepithelial spaces, there are dendritic cells which get infected. They migrate to the lymph nodes. The virus can then infect lymphoid cells in the lymph node as well. It goes to the spleen, establishes a viremia, goes to the spleen where it replicates in more lymphoid cells. And last time or the time before, I told you that TNF alpha is a major reason why this virus gets into the central nervous system. It is sensed by toll-like receptors. The toll-like receptor 3 in particular leads to the production of TNF alpha. That loosens up the blood-brain barrier. Remember the blue brains in the mice last time? And then the virus gets into the central nervous system. So here in part C of this illustration, this is the brain. There's a neuron there on the left, which can be infected by West Nile virus, and that can cause some of the neurological symptoms. And these cells and chemokines and cytokines on the right, these are what are thought to be important for protection against infection, particularly in the CNS, CD8 and CD4 T cells, interferons, and some chemokines. Let's look at a couple of statistics of infection so you get a sense of what kind of numbers we're looking at. This is one that combines total cases over time from 99, that first year when it came to the US, 62 total cases. And it goes up and down over the years, sometimes as many as 10,000 cases. You can see it goes down again. And on the middle line, the top line is neuroinvasive disease, and the middle is non-neuroinvasive disease. Remember, this is mostly asymptomatic, but sometimes you get disease not associated with CNS invasion. So let's see, out of the 62 original cases, 59 of them were neuroinvasive. That's quite a lot. Whereas sometimes it's half or so, or less than half. And these are the deaths on the bottom. So admittedly, this is not a huge death burden, but the neuroinvasive cases are big, and if these people have long-term cognitive and motor issues, they are a burden to the healthcare system. So if you could prevent this with a vaccine, it would be perfect, and that's why we're working on a vaccine. On the left here are the number of cases per 100,000 in the different years from 99 to 2016, just to emphasize that in some years it's very low, in other years it's very high. When it first came to the US, of course, it was limited to the East Coast, but it spread very quickly across the entire country and now can infect many states. And on the right is a graph of the number of cases by the week of onset. So you can see this is a summer disease, right? From July to October-ish. Why do you think this is a summer seasonality? Mosquitoes, that's right. That's a perfect example of how, well, there aren't mosquitoes out there biting you in the winter. They're somewhere, they're hibernating, and they will come back in the summer, but they are not biting you. So there's only transmission there. And this is a graph of, or a figure of cases by county in the US. And the dark is more cases, and the clear is very few. So here in New York, New Jersey area, we have something like a very low incidence per 100,000 people. But you can see in the middle of the US and in the south, and some places in the northwest, there are lots of cases. So normally when there's plenty of water around, the mosquitoes bite birds. But when there's no water, the birds go away. It's dry. The birds go elsewhere looking for water, and instead the mosquitoes bite us. That's one of the theories of why there's a variation from year to year in the number of cases. It tends to go with how dry the climate has been from year to year. And these, overall, this is a 17-year period, the driest parts of the country. And finally, neuroinvasive disease by age group. You can see it increases as you get older. It's the greatest in people over 70. And this presumably reflects declining immune defenses, is a good reason to have a vaccine that would protect you for life. At the moment, the only way we can prevent this infection in humans is by looking out for mosquitoes. These mosquitoes, Culisine mosquitoes, bite from evening to morning. So that's a good time to recognize that either you should have long sleeve shirts and long pants or stay inside. Repellents, window screens, protective clothing are all good ways. There are very many ways to protect yourself against being bitten. And if you're in a place where there are a lot of mosquitoes at that time of year, you should really be careful because although rare, relatively speaking, this can be very serious. As I said, we are developing vaccines. There was already a horse vaccine because they're a lot easier to get licensed to protect them. And eventually there'll be a West Nile vaccine. I don't know who would get this. I guess it would be people in at-risk places, maybe some of those states with the highest incidence of West Nile. Or if you're going to go somewhere where there are lots of West Nile positive mosquitoes, you should probably think about getting that vaccine. Today we have talked about a few viruses that get into the central nervous system. Poliovirus, Measles virus, West Nile virus. They all have a phase where they can get into the CNS, but I think as you've seen, it's pretty rare. And most of the infection leads to other kinds of symptoms by the viruses replicating elsewhere. My feeling is that viral invasion of the CNS is a dead end. It's completely irrelevant to the biology of the virus. Why do I think that? Well, first of all, the main driving force for evolution of a virus is to find a new host. And that's usually driven by high replication in the host and being able to be stable in transmitted situations. And if you get into the CNS, you can't get out. Once a virus is in the brain or spinal cord, it's stuck there. It can't go to a new host. So there is zero evolutionary pressure for a virus to go into the CNS. So that's why I think it's an accident. And the low frequency in these virus infections is a reflection of that. Polio is beautifully transmitted from person to person by fecal spread, by fecal contamination. Getting into the CNS has no relevance to the overall biology of the virus. So the question is, why do these viruses invade the CNS? It's an accident. I think the receptors that the viruses bind to on peripheral cells happen to be also in the CNS, so that's one possibility. But the other is that this discussion applies really to humans, because in animals, you can imagine that there is some selection for a virus to go in the CNS, because animals eat each other. And they eat the brain. Certain animals eat the brain. So you can imagine that virus would be transmitted from animal to animal, even if it went into the CNS. So there would be no negative selection against being in the CNS. And at some point in our ancestry, our ancestors probably did some cannibalism. And so maybe this is a remnant of that period. I think it's unlikely because it should have gone away by now, but it may simply be an accident that can't be prevented. But I tell you this because, Zika virus, when it first emerged, there were a lot of headlines about how it had evolved and it spread from Africa to Asia to South America to become neurovirulent. There is zero reason for a human virus to evolve to become neurovirulent. It's either always been neurovirulent or it's an accident. And in our lab, we have found that every isolate of Zika from 1947 to the present can infect neural cells, can infect brains. It's not a new property, and that makes perfect sense. Now, next time we're going to look at persistent infections, viruses that infect you pretty early on in life and stay with you forever. And there are going to be specific mechanisms that allow them to do that.